So I'm so thankful to introduce Sue Griffin, who's at the University of Arkansas, who's been studying Alzheimer's and had this idea from the get-go that Alzheimer's had something to do with inflammation. And she'll take us through this. We'll talk for about 40 minutes, and then we'll take questions from everybody. So welcome, Dr. Griffin.
Thank you. And I've known Jamie a really long time too. I was introduced to Jamie by my friend Zavan Kachaturian, who was head of programs at the National Institute on Aging at that time. So I've been watching her from a young journalist who was already just top of her game at New.
An old lady.
It's been really wonderful ride, Jamie.
Thank you.
I hope I can do these slides and we can talk about this. So today I'm going to talk to you about Alzheimer's and you. And I'm so happy to have all of you on. And at the one time, I can't see every one of you, but as you speak up after the talk, and I hope you will, then I'll get to see you then. So when you're old but okay, then I want to point actually, and I hope you can see my pointer there, point to the brain here, and I'll give you a little bit of a neuroscience lecture. And I want you to visualize this old guy. He's sitting here in his Barcalounger. You can see he's pretty bored, but his brain is all active because he's okay. So information comes in from his eyes and goes back to the visual cortex here, and he's hearing, so from the ears going into these regions in the temporal lobe. And if you want to know where it is, just point to your temporal, your temple, and you're pointing right at this area here. And this area, when he hears and sees something, then this area is also activated because this is an area of memory and learning. And learning is in large part taking in whatever new information that comes to you and storing it just for as long as you need it. Because we don't need to remember our laundry list from last week. We just need to know what it is for this week and remember it until you go to the laundry and take it and then pick it up. And so all of those kinds of short-term memories are so important for our day-to-day. And we'll talk about that a little bit as the disease process continues in Alzheimer's disease. But these areas like the visual cortex, where information comes in from the eyes, then when you're old but okay, the primary visual cortex here, and then the association areas here where the projections, and then onto sensory cortex. There's this huge area that we still don't know a whole lot about. And that seems to be an area where all of our sensory information is actually processed so we know what to do. And one of the things we know we need to know is we need to know where our parts are everywhere. And so that's sensory cortex. And sensory cortex, of course, is so important because we have to have it to know where our feet are when we stand up. Because if your foot goes to sleep, you know the feeling of not knowing where your foot is. Anyway, and then from there to motor cortex, where we then can put into action what we wanted to do by what we saw and what we heard. And our memories all join together to make us know what we're supposed to do. And what we're supposed to do is in large part decided here in the frontal lobe. That's where we know that, you know, already I'm beginning to think about, oh my gosh, I have to get my tax stuff together. So it's where we do the appropriate thing for the time and the day we're in. And so these areas are very important. And when you're old but okay, and I'm going to tell you about this guy for a little bit. He's watching a movie and he sees this young girl with this cowboy, and he thinks to himself, oh my gosh, that looks exactly like my high school girlfriend. And he just, oh my gosh, that information comes in here. He's seen this image that's reminded him, and wherever our long-term memories are stored, he gets the whole thing. You know how it is. You think of something and you think of a person and you know everything about them. You still remember how it felt the first time you hugged each other. You still remember everything, their smell, their everything about them. And what's really amazing, of course, I've been to my 50th class reunion from high school, so I know this. And it was so amazing to hug these people that I hadn't seen for 50 years. And they're still the same. You know, maybe they got a little fat, but, you know, or maybe they got bald or whatever, but they're still, that person is still inside them. So all those kinds of interactions with the memories in context of what we're
What are
seeing.
you talking about? It's about PCA. You told me you want to watch it. I bothered the person. What is wrong with you?
What's happening there?
I don't know. Let's just keep going.
Hope I don't believe it's on my side, but it could be.
No.
But anyway, so now visualizing him here. So immediately he thinks, oh my gosh, I have to tell someone because this is so amazing. I hadn't thought of this girl in forever. And so he wants to rise up. So he has to know where his feet are. And so then he decides he's going to get up. That's the motor cortex. And then suddenly he remembers that the only person he has to tell this story to is the little lady in the kitchen who's fixing his dinner. And she is not that high school girl that he's remembering. So he just reverses everything and sits back down and enjoys the movie. That's when you're old but okay. Now, how can you be old but okay? And that then is because I want to show you these. These are nerve cells. And these nerve cells here are the things that keep us being able to know where our feet are and to then make actions that we want to do and think thoughts that we want to think. And so one of the things that I can tell you about these nerve cells is that you see them here. They're pretty big. Some of them are not as big as these ones, but this is neuron 1 and this is neuron 2. Neuron 1 communicates with neuron 2 via an electrical signal that goes down to this area called a synapse. It's blown up here so you can see. And so neurotransmitters are hauled down to this area. And when the electrical signal comes, then those neurotransmitters are released onto neuron 2. And then neuron 2 knows what neuron 1 wants it to do. And it does it. And not only that, but neuron 2 actually depends on signals from neuron 1 in order to do well and even survive. So these kinds of connections are absolutely vital for doing everything that we do, from talking like I'm talking to you and looking like you're looking back. So all of that information that comes down, the proteins that are released like the neurotransmitters, are carried along these cable-like things called the microtubules. And that information actually just rolls along as if a railroad track and a car is hauling the proteins down toward the synapse. And that stability of the microtubule is dependent on this little blue tack here that's stabilizing it. In other words, I like to think of it as holding it together. And everything is going just fine at this point, and all the information from neuron 1 gets to go to neuron 2. So nerve cells and how they work then is neuron 1 is speaking to neuron 2. And this is billions. So this is not just 1 and 2, these are billions. And all of them coordinate together to make us do and say the appropriate things. And that happens at this little cleft here. That's the synapse. So we know exactly what we need to do. And here's the nucleus where the DNA is that codes for all the messages that are carried out into the cytoplasm so that the proteins are made. And I'm not sure what those red lines are doing, but anyway, and then that information is carried down through the processes over to neuron 2 for the transmission of information. So when you're old but still okay, everything is fine. But what starts all the things going wrong? Well, one of the really important things goes wrong. It looks like we're having quite a bit of marking on the screen here for me. I hope you're not seeing it. Anyway, one of the things that starts going wrong is inflammation. And here we have inflammation in the joint, which most of us have had experience either ourselves or with friends of the knee joint, for example, swelling up from some trauma. And the immune system that is in our body and the immune system that's really unique, but similarly to the body's.
Well, this isn't working, Leslie.
All these kinds of things are basically to make the joint healthy in the case of the knee and make the nervous system, the nerve
Excuse me,
cells.
we can't hear.
Okay, just could you just just be patient, and I'll fix it once we continue. I'll try to fix it. Go ahead. I'm sorry.
Okay.
Go ahead.
Sure. So let me just point out two of the small proteins that are important both in fixing up the knee joint, and if you obey your people who are helping you with rehab, then your knee will heal faster. But the two of those proteins are called TNF alpha and IL-1. And these two are okay if you remember them, especially IL-1. So what happens in inflammation?
Inflammation in the brain. What does this mean?
Please turn your mute button on, please.
Inflammation in the brain.
Please
Yes.
turn your mute button on, please.
Oh.
Okay. So one of the things that happens is in acute injuries, like you get a very small, relatively small head injury, for example, then, you know, the nerve cell can be repaired. And the way that happens is you get this increase in this inflammatory cytokine that we saw in the knee and also here in the brain. So it's interleukin 1, or for short, IL-1. So when this nerve cell is stressed, it releases a protein that activates this glial cell that's alongside. And here I drew around it just so you can see it. And so when the nerve cell gets stressed, for example, like a small head injury, then it signals to this microglial cell. They're usually very small. That's why they're called microglia. And then when they become enlarged, like they do also in the knee, when this cell gets enlarged, it actually then releases the IL-1 onto the neuron. And when that happens, the neuron responds by making something that it needs. And that something that it needs is membrane repair, for example. And in the brain in Alzheimer's disease, and this is from a case of Dr. Alzheimer's himself, my friend Manuel Greber went there and obtained some slides and stained them with a silver stain so that you can see the big plaques. And these plaques are all in the same thin, very thin ribbon in our brain that has the neurons in it. And so I thought that this is one of the first kinds of pictures I saw when I first became interested in Alzheimer's disease. I was sitting in a lecture and I saw these plaques so close to these neurons, and these are completely inappropriate in the brain. They aren't in the brain except when you have Alzheimer's or some other kind of dementing disease that allows them to be there. But the neuron, I thought, would be stressed, and in response to that stress, the microglia would be activated and it would release IL-1 onto this cell, this neuron, and cause it to make an important membrane repair protein. And the membrane repair protein in neurons is a beta amyloid precursor protein, which gives you a little hint that this protein actually is the source for this plaque. And so it's the precursor protein, but the main function that it has, it's a membrane spanning protein that's necessary for the repair of the membrane of the neuron. That's very important because the neuron membrane, just like any other cell in the body, if the membrane is compromised, then the cell will die. And so this important function that IL-1 promotes in the nerve cell to save it by keeping the membrane intact. And the membrane being intact, of course, is important. And also, in that situation, when everything is okay in the neuron, you have this railroad track-like structure in the neuron that carries the proteins, including the neurotransmitters, down to the synapse for the function of the cell. Well, something different happens in Alzheimer's disease. And this IL-1 also induces this to happen, is over time, you get a lot of phosphorylation of this tau. And when it's highly phosphorylated, it likes itself better than it likes the microtubule. So the microtubule disintegrates. And then you don't have the railroad tracks to carry the cargo down to the synapse. So what happens in Alzheimer's on the clinical front? There are the affected regions, the regions that are usually first affected at different stages of Alzheimer's disease. Now, I'll just take 1 second to talk about posterior cortical atrophy. Now, that occurs back here in visual cortex, you know, from our little lesson on the neuroscience of the brain when we're looking at it. This is visual cortex, visual association cortex too. And these are the areas that are affected in posterior atrophy. And so, but the usual case where most Alzheimer's is I can tell who is talking there. Where most Alzheimer's starts is in this area, inside your temple cortex, the area that's important for learning and memory. And this area, and this is what is so vexing to the caregiver, is this area, when it's not functioning well or not functioning, then the person can't remember things that to him or her are very important and they want to remember it. And so their remedy for that, since they can't remember, is they ask you, the caregiver, and they may ask you 10 times an hour. For example, when is my doctor's appointment? It's next Tuesday at 10. And you only need to read it you only need to remember it until next week on Tuesday at 10. But because you can't remember it all the time, and it's important because it's the doctor, then you will ask the question over and over again, which is so hard and it's a big complaint that caregivers have because it's so vexing after a while because they're stressed already. And then another thing that happens is you start to have dysfunctions in that frontal area, the frontal cortex. And that dysfunction means that you won't be able to make the right decisions. In other words, you won't be able to realize that you are driving your caregiver to distraction by asking over and over. You just know that you need to know. And then as the disease progresses, you begin to lose all from most areas of the brain, that big matrix that we have that allows us to know how to put things together and to put things together in a way that's sensible. And for example, we're going to have Thanksgiving, and Thanksgiving for the grandmother or the mother is so important a day because a lot of times the family is there and their whole motherhood, their whole grandmotherhood, they've been the one that put everything on the table at the perfect time. And there's no forgetting that you forgot to turn the oven on for the turkey, or there's no forgetting that you forgot the pecan pie that you were supposed to make yesterday. So in these kinds of situations, your matrix is lost, and therefore you can't put things together, but you still want to. And so it's very difficult, again, for the caregiver to try to make that happen for you. And then in the severe case, you actually have so many personality changes, because here in the limbic cortex of the brain, you actually begin to lose your ability even to have your own personality. And of course, this is very difficult for caregivers, because the personality of their loved one was so beautiful. And now it's altered in usually very distressing ways. And so then we need to know what increases the risk for Alzheimer's disease. And that risk then we know is brain nerve cell trauma and head injury, epilepsy. And I have to bring up this other thing that happens, and that is that the wars are a big factor here, because the person who suffers the injury doesn't realize that they actually have incurred an injury, for example, blasts. And so I think it'll be a good thing if everybody remembers to wear their helmet, if they're going to ride motorcycles and bicycles, and take care of their head if they're going to think about football. And also, I think it'll be a good thing if we stop having wars. So we expose young people who are young in their lives and have plenty of risks ahead of them, so that they will avoid having Alzheimer's disease. So these lifestyle risks that we are familiar with, like obesity and type 2 diabetes, the lack of exercise and less educational experiences, all things that are going on right now in the United States, those are the kinds of things that you can actually do something about and change the lives, especially of our children. We really have to take care of our children, make sure that they are educated well and know about the effects of obesity and lack of exercise, particularly lack of exercise. And then I want to talk a little bit at the end of my talk about genetics. Mostly, we can't do very much about our genetics, but because we inherit half of our genetics from our mom and the other half from our dad, and so we aren't really in control of that. It would be good if we could pick our parents sometimes more in a more useful way, but we don't have that option. And the inheritance of this big risky gene, this is the biggest risk gene for ApoE, for Alzheimer's, which is ApoE. If you inherit from both of your parents, then your odds of having Alzheimer's disease are really increased quite a bit, like 12 to 15 times over people who have 3-3, most of the population. And so I'll talk more about that at the end of the talk. So these things that increase the risk, one of them, obesity and type 2 diabetes. So, you know, the northern regions of the country are about 25%. But in many states like Arkansas, Louisiana, all across the South, and it's encroaching on the northern states as well, they're at about 30%. And that's going to spread over the country. So by 2025 or 50, if we continue in this way, then mostly in our country, people will be overweight and obese. And those changes, those projections match those for AD risk, so they go together. So finally, in 2016, Newsweek caught up with the scientists, which are now virtually all pointed toward prevention as the new cure. So how about let's get educated to the facts, and I've been telling you the facts right now. So what shall we do? How about let's eat right? Virtually every adult and every pretty much big kid knows what they're supposed to eat. And they know that they need to be eating a lot of vegetables, and they need to eat all the colors. Because even in grade school, kindergarten, throughout their education, they're told this. So we know this. This is not some mystery. And then we should eat fruits. And of course, I have this note for diabetes, because some people with diabetes is so bad that even they even have to limit their fruits. But that's not the case for most people. So we need to eat right, and then we need to exercise. And you know what? It is never too late to start exercising. And you can't believe how much you can strengthen your legs, which is very important, by just exercising. And so it's really great if you can go to a gym, so you can be with a lot of other people. Being together with other people is so important. And then exercising together is even better. And you know what? Great exercise is dancing. Dancing is so great. And of course, it's just so much fun besides. And so you're exercising. And I think if you sing, it even is better. So sing and dance. And this lowers inflammation. And actually, exercise is pretty much the only thing. They've shown even in people with mild cognitive impairment that if they exercise, it seems like it's beneficial. And of course, it's always beneficial to be together with other people, to be able to talk and express your opinions. And of course, you should do this, but be careful. Don't do exercises that are dangerous for you, because you don't want to fall, because you'll have maybe a head injury. And of course, broken legs have set you back. You have to be in bed for a little bit. But get up, do your best, but be careful and thoughtful about what you're doing. But do that exercise. And remember, it's never too late. So take care of yourself, and then take care of your young ones. And this is especially important with caregivers, I think, because most of the time, it's either a wife or a daughter or a daughter-in-law that's taking care of the person. And then there are a lot of women, of men that are also taking care. And in those cases, it's usually the spouse. And so you have to be taking care of yourself, because you don't want to get in a situation where you can't care for your loved one. And then at the same time, you have to take care of them. So it's quite a stress. And we have to take care of our children and our grandchildren to let them know that we care about them, and keep helping them to learn what are the good habits. And then most importantly, to keep loving yourself. And so now I want to turn to this other topic about science and where there is hope, because there is hope in science. And what you have to look toward, even though you think, well, it's just been so long and you've been working on this for such a long time, and why haven't you discovered? Well, I'll tell you why. Because the brain is the most complicated thing that we have in our body. And everything in our body, even just what we think of as ordinary things, like being able to sit upright or to stand, these things are dependent on very complex cycles that are operating at all times in your body, with proteins, signaling, etc., that are complicated. But the brain is especially complicated. And it's especially important because it's so unique, because that's how we know what is the appropriate thing that we should do at all times. And so science discoveries, they may seem small, and mostly I think they are. I admit they are. But each small thing is a part of a huge and giant endeavor to find out how the brain works and how we can make it work better. So it's happening here at the Reynolds in our program in Arkansas. We're now developing drugs to defeat the hyperphosphorylation of tau. And in this way, we can keep nerve cells from forming the tangles, from letting go of the microtubules. So the microtubule falls apart, and forming those tangles of that tau that causes them to lose those necessary functions And then finally, the neuron is lost. And that's a loss then to a complex web of interactions that allow us to do what we want to do and remember what we want to think. And so at UAMS, we tackled one of the problems, and that's the problem of inheriting the risky genes, ApoE4. And if you inherit that gene, the ApoE gene, you can inherit it in 6 combinations. And I won't go over all of them, but I'll tell you the one that's the risky one. And that's if you inherit the sequence that codes for the DNA sequence, that is, that codes for ApoE epsilon 4 from both of your parents, then your genotype is ApoE 4,4, which increases the risk by about 12 to 15 times over that of a person who, say, has ApoE 3,3, which is most of us, by the way, and not very many people have E44, but there's enough that have at least one gene of 4 from one of their parents, and about 50% or maybe even more people with Alzheimer's disease actually have an ApoE4. They have one ApoE4. So it shows you the importance of this. So in our group, we decided to confront that problem and see what is going on. And we knew one thing, maybe quite a bit, we knew one thing, and that is that in the brain of people who are genotype 4,4, there are more plaques outside the nerve cells and more tangles inside the nerve cells. So my graduate student, Paparkan, had this brilliant idea that maybe they can't get rid of the trash in the cell, that maybe that's what's happening. And you know, every cell in the body, whether it's a liver cell, a knee cell, or whatever, has to carry out the trash. They're just like we are. And if you don't carry out the trash, it stinks. And furthermore, it crowds up the cell. And that's what it looks like when you have tangles inside the cell, inside the nerve cell, that is. And so this is the process. Now, when we leave this lecture today, you can say, I should at least have a certificate in neuroscience because I'm teaching you some science. So here we see how proteins that are unwanted, they're trashy and we don't want them anymore. And maybe that's a little bit like, I don't know where you saw your mama do this or you've done it yourself a zillion times, is when you have whatever's left over from the roast or the fish, then you can wrap it in newspaper. And this is some proteins that may be misfolded. And so the cell's got to get rid of them. And I mean pronto, because these sorts of things have a tendency to aggregate. And when they aggregate, then that's what happens that forms the tangles inside the neurons. It's aggregates of proteins. Because when the tau proteins start to interact with each other, they actually function as a garbage collector and other proteins join with them. And so the cell's way of counteracting that is to wrap it, like in a newspaper if it was fish, then wrap it in these little curly things that look like that are actually called ubiquitin molecules. And these ubiquitin proteins then wrap up that aggregate. And when it's fully wrapped up with the curlicues, then these little fish-looking things come over and wrap it up again. And that is wrapped now, the ubiquitin, the trash in the middle, wrapped up with ubiquitin, and then wrapped again with P62. And then that is wrapped once more with this little protein called LC3B. And LC3B then wraps up that whole structure. And then the wrap is complete with all the little LC3Bs intact. And then it can join with a bag of acid that's called a lysosome. And it does that with another protein here that's not written here, but I want to tell you the name of it, is LAMP2. So it's like the lamp says, come over here, and then it joins with the lysosome. And the lysosome is just a big old bunch of acid, and it's released into this structure, the autophagosome, and then that is broken down and the cell is fine. It's carried out the trash, and that's a wonderful thing to happen. Now, you know, I said earlier that every protein has its own unique sequence code in the DNA, and every protein has a promoter region for that region of DNA that codes the message. And so the factor that activates that promoter region, when it lands there, then that unique protein will be translated off as a message, and then transcribed off as a message, and then translated as a protein, and the cell can function. So every protein has a sequence. So interestingly, the DNA codes for the 3 get rid of the trash proteins, LC3B, P62, and LAMP2, reside on a unique sequence alongside each other of DNA called the clear site. And that's, the clear is such a cute name because it clears out the trash, and it stands for the comprehensive lysosome expression and regulation site. So when these 3 messages are laid down from the DNA, then they're translated into the proteins in the cell that are important for joining with the lysosomes for the clearance of whatever you want to get rid of as a cell. Then one day, this is my graduate student, Paul Parkon, and this is Sundram, Meenakshi Sundram, and me, of course. And I'm so lucky. So Paul and I were standing around talking about what would ApoE4 do, and maybe there was some interaction between ApoE4 and that promoter region that starts out activated so that the proteins, the 3 proteins, will be made. And we were talking about it, and Sundram is a bioinformaticist and also a molecular modeler. And he's completely clever, as is Paul. And so when we were talking about it, we didn't realize it, but Sundram had walked in on our conversation, and he said, well, if you're right, and there is an interaction between this protein and DNA, then I can model it for you. And forthwith, over about a year, we did it. And we found that the protein encoded on the ApoE4 gene, it's called ApoE4, competes with the controller that lands on the promoter region for the clear DNA site. So, voila, we thought, okay, I think we got it. So we discovered, all 3 working together, mostly Paul and Sundram, because I stand around and marvel at everything everybody's doing in the lab. So we discovered why inheritance of ApoE4 from both parents confers 10 to 15 times the risk for developing Alzheimer's disease. And you can see it here with ApoE3. This is the protein. This is the double-stranded helix. Here, the protein, you can see with ApoE3, the beautiful image here that was made by Sundram, by the way, and then the interaction with ApoE4. And it's clear that ApoE3 has very little interaction, whereas ApoE4 has a tremendous lot of interaction. And so this is the sequence of events that happens when the transcription factor that activates the expression, the laying down of the message for the translation then of the proteins, that information, the promoter comes into the nucleus and interacts at that promoter region, and If you're ApoE3, which we depict here, or ApoE2, the other choice, which is very rare, if ApoE3 is in the nucleus, then TFEB still can bind to the DNA at the promoter region, and the messages are made, and the translation of the proteins can occur, as we see right here. So here's the promoter interacting with the DNA well, and ApoE3 is minding its own business, and LC3B message, P62 message, and LAMP2 message is made and goes into the cytoplasm for the making of the proteins, and everything is fine. But here, if in contrast, TFEB comes into the nucleus and ApoE4 is in the nucleus, then it will compete for that promoter region on the DNA, and it competes a lot. It's more than, in our experiments, it's more than 50% competing. So your chances of having the ability to make the proteins is decreased by more than 50%. And so you can imagine that this would have a devastating effect on the ability of the cell to clear out the trash. And then you see the buildup of the tangles, you see the dysfunction and loss of the nerve cells. And here I show you, I hope I'm going to show you the interaction. Here's the ApoE. This is ApoE3. You see it in these pipes and squiggly wires. And here, the little part here, the beautiful little part is the DNA. And so this is ApoE3 interacting, and I'm going to show you how it works. And here you can see ApoE3 may have just a tiny bit of interaction, but then finally it's swinging free and not having an interaction to speak of with the DNA. So the DNA is free. And here, in contrast, we see that the interaction here is quite prominent. And I wish I had better finger control so I could show you how it goes. Okay, here it goes. So to me, these two interactions are similar to here in ApoE3. The interaction is sort of like what I think of as today's dancing with each other, where in large part, you may be catching a finger hold once in a while with your partner, but mostly he's just swinging you out into the distance. On the other hand, this is like old time dancing, like waltzing, doing the two-step, etc. All of these kinds of dances, there's lots of contrast, lots of belt polishing going on. And so in these situations, then, you can see how ApoE4 interacts with DNA to interfere with the cell, the neuron, the important thing that we think with and do with its ability to get rid of aggregates inside it, those plaques that occur outside the cell and those tangles that occur inside the cell that foil the neuron's ability to function well. And finally, the neuron is lost. So this all depended on the work in the lab. You've seen Paul and Sundram, and these are other people that contribute and do their parts, from the molecular biology to the cell biology to the physiology. And we have had a lot of help from our whole group. There are 29 of us, 16 are immigrants, so brilliant and so necessary. We must have them. And then our big crew here, and led with by all of us, and then especially Dr. Reese and Dr. Barger and some of me. And so these efforts are sponsored by our program project from NIA that we're now in our 19th year. So we're working hard together with our families here in Arkansas to help us in our efforts to stop Alzheimer's. Thank you so much for coming and being with me today, and I appreciate so much getting to talk to all of you. And I loved getting to watch some of you while I've been talking. So it's lovely to meet everyone. I wish I could meet you one-on-one so we could chat and I could tell you how much I care about not only people with Alzheimer's disease, but trying to find and solve interactions in a better way for you as caregivers. Thank you. If someone has questions, then they should speak up. You have to turn your mics back on to tell me. I see Isabella.
No, I don't have a question at the moment. But
Okay. Then if there is somebody who has a question, just blurt it out, and maybe I can see you, and maybe I can't. Okay, I think that's Norm.
Yes. My understanding is that ApoE4 is not associated with PCA, and is also, PCA is not hereditary. So I'm not sure how this affects us. Our children, for us, we worry about our children with the ApoE4, which we don't have.
Yeah. Well, you know, I'm not sure that the fact that there's not an association, it may be due to two things, Norm. One of them is that the population of people that have both fours is pretty small, maybe 10% or so. But, and then the disease itself is not prominent in the area of Alzheimer's disease. It's looking like there's not a large fraction of the population with that, population with Alzheimer's, I mean. And so those two combinations then would make it where you wouldn't necessarily find it in a population study. But because, and we're studying this further because we want to know whether every cell in the body, cells in the heart, wherever, all of those cells have trouble getting rid of unwanted, misfolded, or aggregating proteins. So I think that it can have an effect, even though it may not actually skew the number of people who have posterior cortical atrophy. Okay,
Any other
anyone else?
questions?
Yes, this is George. I had a quick question. I appreciate the presentation. You mentioned the clear region on the DNA. I was curious whether that exists in animals, whether you're doing like animal studies.
Yes, we are. And yes, it does. And of course, that's where most of our information comes from. Now, we here at Arkansas have a big brain bank of people with Alzheimer's disease and age-matched controls, people of similar age who do not suffer. So what we do actually is we do a lot of cell studies looking at specific cell types in the brain, like the nerve cells and these glial cells that I talked about. And then we also look in animal models of Alzheimer's disease and animal models of just having 3 or 4. And so these kinds of studies, then, we always bring in what's happening in the disease tissue, tissue from people who have allowed us to collect brain tissue for our studies. And so we start from cells, we start from actually molecules, proteins, and then cells, and then animal experiments, and then we see whether or not it has some relevance to what's happening in Alzheimer's disease. So we don't just do those studies and then whatever happens in animals say that must be what's happening in people. We always check to see if there's evidence that's happening.
Yeah, so I guess related to that, I was wondering if you'd reached the point of actually interfering with the clear promoter in animals and seeing if that results in similar kind of behavior, or you're just at the early stages and really haven't gotten to that level.
Well, we do know what's the interaction with just regard to what I was showing you, that, it does interfere, and it interferes pretty smartly. I mean, when you interfere more than 50%, then that's really pretty amazing,
Right.
devastating, I guess. But I think I know what you're talking about, because there are methods now that you can change DNA sequences, but that's not practical, because that would mean that in our ways of doing things now, it would maybe mean transplants, bone marrow transplants, et
Oh,
cetera.
I'm looking at the understanding of it, not for cures for humans.
Oh, okay. Yes.
Yeah, no, I wasn't trying to imply.
Yeah.
I was more just at the getting to understand the biology of it.
Well, it's quite well understood, and that's what made it possible for us to actually ask the question, because people have been very interested in how cells rid themselves of misfolded proteins, because misfolded proteins are very important in a lot of diseases in other parts of the body, not just the brain. And so people have been looking at this process for, decades. And because it's the problem, if you start getting tangled up proteins in any cell in your body, then that cell will misfunction, and you'll have something, like the dystrophies, and et cetera.
Is your theory here that the amyloid is more a byproduct of the APOE4, if I understand it, results in the amyloid plaques, but really it's the APOE4 involvement in interfering with the these clear genes that actually results in the tangles that actually causes the more damaging impact?
Right. Yeah, I think it's very nice that you understand this quite well. And so what we're doing, we are now looking more toward how we can interfere at the level of, say, for example, neuroinflammation, and how we can, which we know a little bit of how to do that, because, the big VA study that was done to see if veterans that had been taking ibuprofen, for example, which is an anti-inflammatory, and they looked over a large group of people, because the VA has a big database. It's been kept really great for many years, decades. And so they looked at 50,000 people who had a confirmed, neuropathologically confirmed Alzheimer's disease, and contrasted them with 200,000 people that didn't have Alzheimer's disease. And they followed them over a 5-year period. And if people had been taking ibuprofen for a 5-year period in this study that was done on the database, their odds of being in the Alzheimer group was decreased by about 43%. And so that's a way for decreasing. But then there are all the reasons why maybe a person wouldn't take ibuprofen at the levels or whatever, because they were taking ibuprofen for not for their Alzheimer's disease, but for their aches and pains. So, so we still don't know exactly what's happening there. But we do know this, that one of the ways, now that we can do this molecular simulation, then we can pick out specific targets, which we are doing, on these proteins, that we can direct drugs. We can make drugs that fit into those regions, and that then in that way we could actually decrease the amount of that protein that's being made. So that's a way that we can interfere in this process. So we're taking that kind of approach, using the tools that we have in our group to actually counteract the making or the misdo of proteins in Alzheimer's disease that are causing the problems.
May I interject a question?
Yes.
Or do you have enough time? Sure. I heard you touch on the word inflammation, which is related to my question. In your lab and with your group, are you at all interested in what has reemerged as considering what's been known, I guess, as the germ theory?
Yes, I just came from a meeting. I'm just interrupting you a little bit.
Yeah, you know what I'm talking about.
From a meeting on this topic, and it is a hot topic now, and people are worrying a lot about it. And of course, it's a bunch of people have, for example, viruses that cause cold sores, And so, you know, those kinds of interactions, it's like, oh gosh, I really hope that that's not a big problem in Alzheimer's disease. But I do know this, any way you stress the neuron, if it's a virus, and our lab has shown each one of these kinds of hits on the neuron. If it's a virus like HIV is what we looked at, and if it's a head injury, which we looked at in like motor vehicle crashes where a person survives for a short period of time, like 12 hours or something. And then in epilepsy, where we've had several large studies on epilepsy with and without ApoE4. So each one of these kinds of stresses on the neuron sets up this cycle, which is to speak to this inflammatory cell, the microglia, and there are astrocytes, also the other cells that can do this too, releasing IL-1 onto the neuron, and that starts the cascade for the making of the beta amyloid in the plaque and the making of the hyperphosphorylated tau in the tangles. So this inflammatory cycle actually is contributing to the laying down of the plaques and also the laying down of the tangles. So it's very important. And exercise big time, eat right, teach children. I think those are the messages that I want to convey to you. And I want to thank you, each and every one, for being here. And I wish you the very best.
Thank you so much.
Endeavors.
Thank you. I have one question, if you can hear me.
Yes, I can.
So I've done a lot of stories recently because inflammation is a really important part of Alzheimer's, and a lot of people are paying attention to it. Do you think it's worthy of getting, you know, a simple blood test for C-reactive protein, which is a marker for inflammation, to see if it's high, or do you know what I mean?
Yes, I definitely know what it means. And I was sitting in a seminar, and it was right before the seminar began, next to this guy who almost 2 years ago, when I said what we really need to have is a neuroinflammatory test that will actually let us know whether or not these proteins are being expressed. And those are the very ones like C-reactive proteins, the cytokines like interleukin-1, TNF-alpha, and I was talking about that in an address I was giving to a group of people, and he said, "Okay, I'm going to make it for you," and he hadn't done it yet. So I was sitting by him day before yesterday. So I was telling him, "What is this that you told me all that year and a half ago that you were going to make this for me, and I still don't have it?" So I was putting the B on him. And he said, "Oh, well, I'm going to do it now." So I'm hoping in no time flat he's going to do it, because he's an expert on making these kinds of complexes.
Wonderful. Any other questions before we take off here? Okay,
yeah, do you have any opinion on taking anti-inflammatories at this time? I know they're doing a study at UCSF with salsalate.
Yeah, well, you know, I would get in a bunch of trouble because I'm not an MD, but there's some days I wish I could be, but not really. I told you about the study from the big database, 50,000 people with confirmed in the clinic and confirmed in neuropathology. In other words, they had enough plaques and tangles to say that yes, the clinical diagnosis was correct. So 50,000 of those compared to 200,000 age-matched controls that neither had clinical signs nor neuropathological signs. And so in that study, it looked good for ibuprofen, but, you know, I don't know. I guess just like every day, we have these decisions that we make. And there are warnings for everything we take. There's warnings for every step we take all day long. oh, be careful. And we just have to decide which be careful we're going to say. And one of the things that's really important to say is don't go falling. Don't hit your head. Okay. Those are the important ones. And get that exercise. Enjoy each other as much as you can.
Perfect. What a nice way to end a terrific meeting. So I thank everybody for coming here, and we're going to get this up online later. And thank you, Dr. Griffin. You were terrific.
Thank you, everybody. Happy Thanksgiving.
Yes.
Thank you. Thank you all.
It was great.
Thanks, everyone. I'm so glad I could see your faces.
Bye.
Bye.
Boy, that was great. Hello.
Hey, Jamie, can I ask you a question? Or are you gone? Okay, bye-bye.
Jamie's still on.
Oh, do you have any, are there any other groups
Sorry,
besides?
I'm not Jamie. I'm not Jamie, but she appears to be connected somewhere.
You know what? Everybody has to kind of hang up because my computer is not letting me. So.
Okay. Can I ask a question, Jamie?
Sure.
Is there another place that we can get information besides the Facebook group? Because I'm getting a little tired of Facebook.
Okay, so I have a website called PCA-vision.org, and you can get information there. I have lists of doctors who study PCA. I have, all of the information about what PCA is and contacts for clinical, doctors across the country. And you can always, if you have a question, you can always email me.
Okay, thanks.
And we have our monthly support group for patients and then for caregivers and patients as well.
Well, it's good to hear from other people too, though, what they're going through.
Exactly. Well, come to the support groups.
Okay.
Thank you, Norm.
Thanks. Bye.